Further in diatomaceous fungi, the Fonseca campata, now considered as a mutant from the Fonseca pedrosi, is a major cause of chromoblastomycosis, that is skin and subcutaneous tissue characterized by the pseudo epitheliomatous hyperplasia with the hyperkeratitis or parakeratitosis. Very slow and mature in 28 days. Surface is dark green to black and brownish. Hyphae reverse is black. The outstanding characteristic for this species are the cast-like shape of conidia and the compact arrangement of the conidia chain. Chlorostomophora richardsii is formally also called Pelophora richardsii, is an uncommon agent of cystic subcutaneous pilohyphomycosis, most common in the skin and also occur in the brain and occasionally in the variety of other body sites. Severe disseminated infection has also been reported. It is moderate, mature in 6 to 10 days. Surface is olive brown to brownish gray and the reverse is black. And another organism in diatomaceous fungi are Rhinocleidelia species. Some species were formerly Archoteca species, others were Rhinocoloridium species, and several species have been occasionally been involved in the chromoblastomycosis and mycetoma. The Rhinocleidelia McNeesiae has repeatedly been reported to cause the fatal intracranial infection in the Middle East. The moderately rapid to slow, depending on the species and surface of the colony, may smooth, velvety or woolly, dark gray to greenish black, and reverse is black. So, surface colony of Fonseca pedrosi. Reverse colony of Fonseca pedrosi. Philophora. Chlorostomophora richardsii and Philophora, basically same species. Philophora versuca cause the chromoblastomycosis, is the second most common etiological agent worldwide, and also cause the hyphomycosis, moderate and mature in 7 to 12 days. Surface is black, dark greenish brown to black, and the reverse is black. Hyphae are brown branch and septa, and pilides are visa shaped with the flared cup-like colorate and giving the appearance of a visa flowers. Almost the surface and the back colonies of on the culture plate is same for all of these three species. We can only differentiate it on the basis of its morphology on chromothiamine blue staining. Cladosporium species also categorized in diatomaceous fungi. They are occasionally cause the infection, moderately rapid 7 to 10 days at 25 degrees centigrade. Surface is greenish brown or black, reverse is black. The conidiophores form the tree-like chain and are easily dislodged and showing the dark spot helia, which is the basic you can say differentiation or unique property of this Cladosporium species from the rest of diatomaceous fungi, resembling the shields. That why it is also called the shield cell. See the table. If you see the table, another important organism in diatomaceous fungi are Pseudoerythia bodii is a sexual state, and Scidosporium bodii is a asexual state of this fungus. It is a common cause of mycetoma, and mycetoma is subcutaneous tissue or a skin or long standing infection, may involve the muscle and fascia and bone, and the infection is most commonly on the lower leg or foot, rarely disseminated. This is moderately rapid, mature in 7 days. The asexual state grow on the media containing the cyclohexamide, but the cleistothecia of the sexual state are often inhibited. And the surface has a spreading white, later turn gray or brown. Reverse is first white but usually become gray or black. For further differentiation, we can check the table, but the most important is the picture of this organism. We can check the picture here. Surface colony of Pseudoerecia boii. Coroconosis gallopoa is formerly Dactylaria constricta, has caused the subcutaneous and disseminated infection in immunocompromised. It is common cause of encephalitis in the turkeys and chickens and other birds. Rapid, mature with 5 days. Growth is inhibited by cyclohexamide. Surface is brownish black and reverse is black. The structure is quite different, so we can identify this organism on the basis of its morphological culture characteristics. Exophilia gensilemae complex have also different structure morphology. It is rarely reported but can cause the phylohypomycosis, chromoblastomycosis, and mycetomas. It's a slow growing, mature in 14 days. The surface is brown, black, or greenish black. Reverse is black. Cornmeal agar, potato dextrose agar may have many yeast like cells with the septate hyphae. Exophilia dermatitis causes the phylohypomycosis but can be disseminated to the central nervous system, lungs, and eyes. Slow growing, mature, filamentous within 25 days, and yeast like in the 10 days. And at the first, the colony is black, moist, shiny, yeast like, and reverse is black. Angkora gulpo on SDA after 7 days. Exophilia gensilemae complex 14 days. Surface colony. Exophilia gensilemae complex SDA 30 degrees centigrade 14 days and reverse colony. Exophilia dermatitis on SDA 30 degrees centigrade 21 days. Surface colony. And Exophilia dermatitis reverse colony after 21 days. Horti wernicki is formerly called the Phonilomyces wernicki, an etiological agent of Artemia nigra, a superficial asymptomatic fungal infection of the skin and usually on the palm of the hand, and the lesion are flat, smooth, and not clay. Develop slowly, mature within 21 days. Surface is first light yeast color but soon become olive black. Reverse is black. The iniloconidia may form and accumulate at the inilid and produce the new conidia, and chlamydioconidia may develop with the age. In diamintinous fungi, Medurola mycetomitis is a cause of black grain mycetoma, most common in the South America, India, and East Africa. Moderate, mature in 10 days. Best grow at 37 degrees centigrade, much slower at 25 to 30 degrees centigrade. Various greatly powdery, range in the color from the white to yellowish brown. Reverse is dark brown. On SDA, form the septate hyphae with the numerous chlamydioconidium like in large cell. M. mycetoma differ biochemically from Medurola grisae in assimilating lactose but not sucrose. Pedria horti is caused the black pedria, a fungal infection of the hair of the scalp. It is characterized by the formation of a small, stony, hard, dark nodules along the hair shaft. And the white pedria is caused by the Trichosporum ovoides and Trichosporum incans in cetol positive yeast-like organism. Basically, Pedria horti is a slow mature in 21 days. Surface is dark greenish black and reverse is also black. The hyphae are closely septate and dark and thick walled with the many intraciliary chlamydioconidium like cells. Ascospores are tapered at the end to form the weep like extension, and the ascospores are more likely to be seen on the direct microscopic examination of the specimen than on the culture. If you see the ascospores here and the colony, the mycelium hyphae properties are quite different from rest of the species, so that it can easily be differentiated. This is also called the black P3. New Cystolidium diamidatum. New Cystolidium diamidatum is commonly caused the nail and skin infection. There are also rare reports of a more deep-seated infection, for example, subcutaneous abscesses, sinusitis, endophthalmitis, and lymphadenitis, brain infection, fungemia in immunocompromised patients. Rapid mature with 3 days growth is inhibited by cyclohexamide. Colony are usually woolly and growth quickly fill the agar plates. The surface is grey to brown with the dark reverse hyphae are septate branch, but no conidiophores are formed. Carvolaria species also characterized in diatomaceous fungi. It is opportunistic. In fact, opportunistic pathogen most commonly cause the cornea or sinuses or mycetoma or pilohyphomycosis. It is rapid mature within 5 days. Surfaces are olive green to brown or black. Woolly surface reverse is dark, and conidia differ from those of Biopolaria species by having the central cell that is darker than the end cells. Bioporellus species most commonly cause the allergic sinusitis, and in immunocompromised patient many progresses to involve the bone and cause the lesion in the brain, and rapid mature within 5 days. Surfaces at first grayish brown and become black, reverses dark brown to black, and dark septate hyphae conidiophore elongate. This produce a novi zigzag appearance when tested from the green germ tube production. The germ tube develop at the both poles of the conidia, hence this genus is called Biopolaris species. Corvillora species on SDA 30 degrees centigrade after 5 days. Biopolaris surface and reverse colonies. Xerohilum species and Alternaria species, which we will first further explain. Xerohilum species most common in nasal sinuses, skin, subcutaneous tissue, and cornea, and fetal disseminated infection has been reported rarely, and the rapid mature within 5 days. Surfaces dark grayish to black, cottony, and reverses black, and conidiophore enlarged novi zigzag, usually 7 to 11 septa is there. If you see the germ tube, in the germ tube, from the one pole the feet appears, which differentiate this organism. Alternaria species are occasionally cause the pilohyphomycosis and few cases of infection of a nail's eye and nasal sinuses. Additionally, it may play a role in asthma and hypersensitivity, pneumonitis, and it is a rapid mature within 5 days. Surfaces at first grey white and woolly, later become greyish black, reverses black, hyphae are septate, and dark conidiophores are septate, and producing the club like shape. They have both transverse and longitudinal septation, and sometimes produce the germ tube, usually in change, but singly also can be found. If you see, the structure of Alternaria is quite different from rest of diatomaceous fungi. Now we will start the hyaline hyphomycetes. In hyaline hyphomycetes, we will explain the first. We will define this organism. These are the molds having the colorless septate hyphae producing the conidia that may be colorless, pigmented, and the colony surface are white, gray, tan, yellow, pink, or green, and the reverse is white or lighter pigmented. And some of these organisms are known pathogen. For example, coccidiod emitis and coccidiod pityasi. But most of these are opportunistic organism and cause diseases only in immunocompromised patients. Coccidiot species is consist of two separate species. One is Coccidiot emitis is a primary in since Joaquin Valley of southern California and has occasionally been recovered in the Mexico. And Coccidiod paradosi is more widespread, found in the endemic area throughout the America, and the only known phenotypic difference is the ability of aceiomyces to grow more rapidly than C. pyridosi in high salt concentration. It causes the coccidioidomycosis, a highly infectious disease of respiratory tract, may be disseminated moderate, mature within 10 days, and colonial SDA gray reverses the white to gray on a special conversion medium with the increased carbon dioxide at 37 to 40 degrees centigrade. The spherical or a tissue phase form is formed. This is not routinely performed in the clinical laboratory, and it is the arthrochonidia, highly infectious, and this is the spherical form which contains the spores, mostly found in the vitro. Aspergillus is also categorized in hyaline hyphomycetes. Aspergillus causes aspergillosis, colonization, toxicosis, or allergy. The hyphae are septate, unbranched. Conidiophore arise from the specialized foot cells, usually rapid, mature within 3 days. Some species are slow growing, and the surface is white, then any shade of the green, yellow, orange, or brown or black depending on the species. Reverse is usually white and goldish or brown. The conidiophore are enlarged at the tip and forming the swollen vesicle. Vesicles are completely covered with the flask-shaped pilides called strigmata. If we see the structure of the Aspergillus species, you will find the metola to which the conidiophores are attached. And these there is another structure that is called pilides. Pilides are attached to the metola, and then the conidiophores are attached. This property is called biacerate pilides. And if you see, the unicerate pilides are also found in which the metola is absent. Aspergillus fumigatus is the most common cause of invasive disseminated aspergillosis, allergic and fungal sinusitis. The septate hyphae, conidiophore, and pilides are unicerates, which differentiate this organism from the rest of the species of aspergillosis. The surface is velvety and powdery, and various shades of green and reverses white to tan. And note the Aspergillus lontolus is phenotypically resembling the A. fumigatus. However, it have a slow spolurating stance to be more resistant, does not grow well at 48 degrees centigrade, while A. fumigatus does, and occur uncommonly among the clinical isolates. Aspergillus niger is most commonly isolated from the external air and causing the osteomycosis, and also a frequent agent of a fungus ball in the pre-existing pulmonary cavities. Only occasionally causes the disseminated diseases. It is rapid, mature in 3 days. Surface is black with the white border and reverse with the white cream, and septate hyphae, conidiophore long, smooth, and cluster of the ball conidia at the apex. Bicerate metola twice as long as the phylides.